After coming to you last month from Ag Progress Days, we're back on Ag Hill for today's College Connections. I'm Rick Roush, Dean of the College of Agricultural Sciences, and I am, as always, delighted to welcome you to our College Connections monthly webinar series. This series is designed to give you a unique inside perspective of our program, people, priorities, and partnerships of the Penn State College of Agricultural Sciences. This is the 8th session, and we hope that you're enjoying these opportunities to get an inside look at all that's going on in the college. Before we begin, please note that we are recording this session and we'll be sharing it via email with all who registered. If you know someone who may be interested in what we cover here today, we encourage you to share the recording with them. We have a dedicated webpage, which you can find by searching PSU College Connections. You will also find links to all of the past sessions, as well as registration information for future events. Today's topic is modern genetic approaches to advance agriculture and human health. This has been a topic of personal research interest for me. From 1994 to 2005, I authored or co-authored 12 papers on methods to delay the evolution of resistance in weeds and insects to genetically modified crops. Where the recommendations of these papers have been followed, which is mostly in the United States and Australia, the evolution of resistance in insects and weeds has been far less than in countries or regions where the recommendations were not followed. I'm joined here today by two scientists who've worked in very different areas of genetic approaches to agriculture and health, Dr. Jason Raskin, professor of entomology and disease epidemiology, and Dr. Mark Guiltinan, the J. Franklin Steyer Professor of Horticultural Botany and Professor of Plant Molecular Biology. After their presentations, we will be addressing questions that were submitted during the registration process. Also, please feel free at any time to enter any additional questions into the Q&A link, and we will address them as time permits. To kick today's sessions off, I'm excited to introduce Professor Jason Raskin. The Raskin Lab, also known as the Vector Genetics and Control Laboratory, works at the interface of mosquito genetics and vector-borne disease control. Today, he'll be discussing research on genetically modified mosquitoes and their use for disease control and pathogen transmission. Jason earned his bachelor's degree in zoology from San Jose State University, his PhD from UC Davis, and conducted postdoctoral research at North Carolina State. So with that, over to you, Jason.
Okay, thank you, Rick. Thanks for the opportunity to present here. So I'm going to share my screen here, see if this actually works. Is that good? All right. So today I'm going to be discussing vector-borne disease control within the context of the new genetic transgenic methods that have been developed over the last decade or so. And I'll be highlighting not just our work, but sort of an introduction to what's going on in the field in general. And this is going to be quick. I'm going to glance over a lot of stuff because I don't have time to talk about it. But hopefully this should give you an idea of what this technology is about and how it's actually being applied in the laboratory and the field today. I'm going to talk a lot about CRISPR, so I just want to give a very brief overview about this. CRISPR is a technology that within the last 10 years or so has really revolutionized pretty much all aspects of at least biology and life science, and vector-borne disease control is no exception. It's at its bare minimum is a system that allows precise editing of DNA. And all that CRISPR really is a way to make a break in a double-stranded piece of DNA at a place where you want it to be. That's all CRISPR does, and it's very basic. And then it exploits the endogenous cellular processes to repair these double-stranded breaks. Cells don't like to have double-stranded breaks in their DNA. It's bad. So when one of those happens, the cell will repair it. And this repair process tends to be error-prone. And so by exploiting that error-prone nature of the process, you can damage a gene essentially by cutting it. And when the cell repairs it, it'll make a mistake, and then that gene will no longer be viable. This is what we do when we use CRISPR to knock out a gene. You can also sometimes the cell will insert genomic material into that cut site to repair the break. This is how we use CRISPR to insert genetic material into the genome. But really, it's just make a cut and repair it. That's all CRISPR does. And then this processes can be exploited also for something called gene drive, which I'll talk about in a little bit. It's important to realize that every single thing we can do with CRISPR right now, we've already been able to do it for at least 20 or 30 years using other methods. The power of CRISPR comes that it essentially has exponentially sped up how quickly you can do things. It's much, much easier than previous technologies. So nothing at its basis that I'm going to talk about is new. It's the application that's new, because CRISPR allows us to do things much easier than we could previously do it. Just a quick plug for our own work. Performing CRISPR in multicellular organisms can be sometimes a bit of a pain because you have to actually engineer the hereditary germline in order to have those genetic modifications inherited to the next generation. My lab has developed a new process called remote control, which, without going into details, is a way to very easily make germline edits in pretty much any organism of choice. And this has been done in a lot of papers. We have patent pending on it. It's worked in over a dozen arthropod species so far, including multiple vectors of human and health and agricultural importance, including multiple mosquitoes, ticks, kissing bugs, and also agricultural whitefly pests. If anybody has any questions about that, I can talk about it later, but I don't really want to focus on this. I'm just sort of putting it up there. This takes the ease of CRISPR and makes it actually even easier to apply it to your organism of choice. So when we talk about vector-borne disease control, we're really talking about two types of control. And this is true whether we're talking about genetic modification or we're talking about traditional control strategies. There's population suppression strategies where we want to reduce the population level of the vectors below some level required for transmission. So an easy way to think about this is an insecticide program where you're just spraying insecticide is a population suppression technology. In terms of genetic modification technologies, we're going to talk about the transgenic sterile insect technique, or SIT. There's also something called the Wolbachia incompatible insect technique, which is functionally similar to sterile insect, it's just by a different mechanism. And then gene drive suppression, where you can push a gene through a population and then crash it. The other type of control is population replacement, where you actually replace the natural population with one that's less able to maintain a pathogen transmission cycle. So genetic modification of mosquitoes to make them unable to transmit malaria. If you could push that gene through the population, you can stop malaria transmission. So gene drive of refractory mosquitoes or Wolbachia invasions. Today, I'm only going to talk about three of these: this transgenic sterile insect technique, gene drive suppression, and Wolbachia invasion, because I don't have time to cover everything. Also, just to say, gene drive and Wolbachia, depending on how you use them, can be used for both suppression and replacement. It's all in how it's used. The sterile insect technique, or sterile insect release technique, is when you sterilize usually male insects and release them into the wild. They mate with the wild females, and then because those males were sterile, those females don't have any offspring. This is a very old technique. It was used in the 1950s at least. Probably and conceptualized even earlier than that. So it's been used for a long, long time. It's very old. Traditionally, we've used radiation or sometimes chemosterilants, but usually radiation to induce this sterility. And this caused problems because when you irradiate insects, they generally aren't very healthy. They don't mate very well, which means you have to release, let's say, 1,000 to 1 or 1,000,000 to 1 in order to have significant impacts on the population. So there are people and companies now that have been using transgenic technologies to artificially create sterile male mosquitoes in this case, or some other species, because you don't have to irradiate them. And so this is done to mitigate the fitness effects of radiation. And one company that's been doing this since the 1990s is Oxitec, where they have these genetically engineered mosquitoes that are sterile. They release them into the wild. And the interesting thing here is, at least with their newer generation of mosquitoes, it's only the females that are lethal. So the males, the transgenic males, are fine, they reproduce, but if they have female offspring, those female offspring are dead. So you can actually get multiple generations of control with a single release. So it's actually much more efficient than traditional radio sterility. And so this is just sort of the idea. You start with a mosquito population, you start releasing these over time, and over time the population will be reduced. And they've actually done this in a number of different field sites. So this is what the first instance of a genetically modified mosquito being released into the wild for disease control. And as long as you can maintain these releases, it's actually very, very effective. Out of all the transgenic sort of strategies out there, this is the one that I like. It's the one that I think is the safest and has the most minimum in terms of environmental impact. It's very, very specific and very, very safe. But the heart of the idea is you are making sterile insects, in this case by transgenesis, and then releasing them into the wild to crush the population. Another way to do this is gene drive. And so again, this is how CRISPR gene drive works. On the left here, you have your CRISPR allele. So you have your Cas9. The guide RNA decides where this thing cuts, and then the cargo could be a gene that makes mosquitoes unable to transmit malaria. It could be something that kills females. Doesn't really matter. The Cas9 will cut the opposite chromosome. Mosquitoes have chromosomes in pairs. And then the cellular repair machinery will use this chromosome as a template to repair the cut chromosome, and it'll copy itself over. So an individual that's heterozygous has one copy of the gene, will have two. And so instead of having normal Mendelian inheritance, you have gene drive inheritance where the altered gene is preferentially inherited to the next generation. And very, very rapidly, this can cause the gene to spread through the population. And now people have talked about using this to spread anti-malaria genes into populations, but really the technology that's most far advanced is what's called suppression gene drive. In this case, it's not a gene that causes the mosquitoes not to transmit malaria. It's a gene that actually kills females. So if the male has it, it's fine. If the female has it, the female's dead. And I'm oversimplifying a little bit, but that's how this works. So when you release these over time, the gene frequency, the frequency of that introduced gene increases. And you can also look at this. This is the frequency of males in the population. So over time, the sex ratio of males goes from a 50-50 to, let's say, 99 males for every female produced. And so as that gene increases in frequency and the males increase in frequency, the population levels go down because the number of females in the population is lower and lower every generation. And ultimately, this will crash the population because you don't have enough females to maintain the population from generation to generation. Again, this is an old idea. It was actually first sort of conceptually developed and even experiments carried out by Cerebrowski and von der Planck back in the 40s, using very different methods, of course. But it's not a new idea. None of the ideas that we're talking about when we talk about transgenic vector-borne disease control, none of them are new. They're all old. It's just an application of a novel technology to these old ideas. There are some pretty big issues with suppression drive, at least in my opinion. It is currently being trialed in semi-field large cage environments in Africa. And the proponents of this are gearing up to try to do some field releases in the near future. The suppression drive distorts sex ratios towards males. So you end up not having females, you only have males. And as the gene increases in frequency, you get more males, less females, and the population crashes as the females become scarce. I think there's pretty good evidence to say that the evolution of resistance to this drive will nullify the efficacy. Because naturally occurring sex ratio distorter genes are incredibly common in mosquitoes. You never see them though. You have to specifically look for them. You have to do certain kinds of crosses because as soon as they become common in the population, resistance genes evolve to suppress them, which is what you would expect. So the frequency of males goes up, the resistance gene evolves, and then it comes back to a 50-50 sex ratio again. I don't think there's any reason to think that these engineered sex ratio disorders will be any different. I have arguments with these people all the time, but it is something that's probably going to be trialed within the next 5 years. And then last couple of minutes, shifting gears, I want to talk about Wolbachia. And Wolbachia is often talked about as a non-GMO strategy, but I don't agree with that. Wolbachia is a maternally inherited bacterium. It's inherited from mother to offspring. And researchers showed when you took Wolbachia out of Drosophila, fruit flies, and put them into mosquitoes, Aedes aegypti mosquitoes, those mosquitoes became unable to transmit dengue virus. And so you're actually taking 1,500 genes in a self-replicating construct out of the fruit fly and putting it in the mosquito. So I would consider that a GMO strategy. But this dengue blocking in Aedes looks pretty good. And Wolbachia, because it's maternally inherited and it also messes around with reproduction, it's able to spread itself rapidly through the population. And so this is how that works. over time, you keep releasing these Wolbachia infected mosquitoes, and pretty soon the entire population is infected with Wolbachia. And then if these are Aedes aegypti mosquitoes, you've taken mosquitoes that can maintain dengue transmission cycle, and they become unable to do that. So you stop dengue virus. This is a technique being driven forward by the World Mosquito Program in Australia. They've actually done field releases all over the world. There is some decent data actually from the Indonesia site where they actually were able to do a randomized control trial, and they did show some fairly significant reductions in dengue virus. So it seems promising. But again, like everything, there are some problems. This is actually what my lab primarily works on, is what happens when Wolbachia doesn't block pathogens, because it doesn't always block pathogens. Sometimes it can actually make pathogens worse. It can enhance pathogen infection or transmission in the mosquitoes or in the other organisms. And that's very sort of dependent on the parameters, the Wolbachia strain, the mosquito, and the pathogen. You can have differential effects on different pathogens. So it can block one thing, but enhance something else in the same mosquito. There's environmental variation. Lab studies might not detect instances where Wolbachia can fail. This is just a list of some of the papers showing that Wolbachia can actually enhance pathogen infection in a variety of different organisms. And on the right is data from my own lab where we took different viruses, different mosquitoes, and different Wolbachia strains. And we can see that sometimes Wolbachia does significantly suppress virus, but other times it actually enhances it. And it's very dependent on which mosquito, which Wolbachia, and which virus. So these interactions are highly complicated. Turns out the mosquito host genome, the microbiome, the other bacteria in the mosquito, and the environment all interact with Wolbachia to modulate virus blocking. Here's an example where we took Wolbachia infected mosquitoes and we used CRISPR to knock out a single gene in the mosquito genome. And when we did that, Wolbachia no longer blocked dengue virus. So just by knocking out one gene in the mosquito, we were able to completely eliminate blocking ability. If you change the microbiome of the mosquito, it turns out you can make blocking better or worse. So Wolbachia interacts with the endogenous bacteria in the mosquito, and this can affect whether it can block viruses or not. And then this is other data showing that when we reared mosquitoes, Wolbachia-infected mosquitoes at different temperatures, you could actually make them better blockers, worse blockers, or have no effect. And how these things will impact control efforts, we don't really know. And so with that, I'll just end there. And that this research has been funded by a lot of different people, and too many people in my lab and our collaborators to list, but there's dozens and dozens and dozens of people that have done this stuff over the years. And with that, thank you very much. I'll end there. You're muted, Rick.
Thanks, Jason. Yeah, hopefully that's got everybody fired up for questions. But first, we're going to go on to Mark Guiltinan, whose research explores the molecular basis of plant development and application of plant science to crop improvement. Today, he'll be sharing information on the identification of key genes for disease resistance, specifically in relation to Theobroma cacao, also known as the cocoa tree. Since 1995, Mark has been the director of the Penn State Endowment for the Molecular Biology of Cacao, chairman of the International Group for the Genetic Improvement of Cacao since 2003, and serves as the director of the Plant Science Center of Penn State's Huck Institute of the Life Sciences. Mark earned his bachelor's degree from Humboldt State University and his PhD from UC Irvine. Prior to joining Penn State faculty in 1991, he conducted postdoctoral research at Texas A&M and at the University of North Carolina, Chapel Hill, as well as within industry. So take it away, Mark.
Okay, thanks Rick for the introduction and Jason for the background on CRISPR. That'll make my job a little easier. Let me start my share up. How's that? Looks good. Okay. All right, so thanks again everybody for coming, and today what I'd like to talk about is a little bit more about the modern genetic approaches to advancing agriculture and specifically crop plants. So we're going to talk about the modern methods of crop improvement, but first I'm just going to give a little bit of a background on the history of crop improvement, and then focus on, give you just one example similar to what Jason did of some of the work going on in my lab. Oops. Yeah, crop domestication has been in plants and animals have been going on since about 13 to 15,000 years at least. Just by human selection of traits that were beneficial for agriculture, as opposed to natural selection, where all the organisms evolve to fit into the natural world. Humans have been domesticating crops and animals for a long time, but more recently we turned this more into a science. In crop plants, one of the big discoveries was the discovery of hybrid seeds, where almost all the corn varieties now are hybrid seeds. And there's a phenomenon known as hybrid vigor. It's basically just crossing two inbred lines of corn together, and you get a synergistic impact of the genetic background of both of the parents. We kind of called this that was part of the first green revolution that also involved the use of fertilizers and pesticides and other things. And also the field of genetics exploded during the 1900s. And, lots of much more sophisticated knowledge about how genetics works were basically used to develop breeding technologies that were much more advanced over the years. But we've kind of come into a very new era over the last decade or two, and that is huge advances in genomics. That is the study of all of the genes of a genome, in the actual discovery of the specific genes that are important for traits. This has enabled us to start to breed much more efficiently for especially complex traits. You heard about GMOs a little bit there. We got our first GMOs in the United States in the mid-90s. Genetically modified organisms, or now, as we now call them, bioengineered. And more recently, as you just heard, CRISPR and other methods of genome editing have come along. We'll talk about that a little bit. One of the exciting new impacts of this is moving this sort of technology into Africa, and I'm not going to have time to talk about that today, but there's a lot of really great applications. For example, drought resistance maize that has been moved into Africa, some plants that have higher nutritive value, and so on. Some people are calling it the second green revolution. Let's see if you agree with that or not. But just to show you, this is some fun pictures of the ancestors to our modern crops. Without plant breeding, this is what would be in the local grocery stores. But instead, now through plant breeding, we have more varieties with better yields, better growth under different kinds of stresses, more healthy and more safe products. As Jason already gave the background on this, I was going to talk a little bit about what CRISPR does, but he already did a great job on that. But one thing I want to point out is kind of the difference between, you heard a GMO or a transgenic organism, and I want to explain briefly the difference between that and a genome edited or a CRISPR modified organism. In a transgenic crop or a transformation, we actually take a gene, and this example shows a gene for disease resistance coming out of a watermelon being spliced into a tomato. So you actually have a chunk of DNA from one organism going into another organism. It's stably incorporated into the genome. It's passed down through seeds into the progeny of that organism. It's stably part of a new GMO. And as opposed to that, CRISPR-Cas9, as Jason pointed out, is simply a cut and a paste, or sometimes just a repair mechanism. You can see that there's no foreign DNA coming into the new organism. It's genome edited. So it's basically like creating a small mutation. Sometimes those mutations can change gene function. Most mutations do change gene function in a way that's bad for the plant or even bad for agriculture. So some mutations, however, may be beneficial for agriculture, and those are the ones we're interested in. And that's one of the biggest challenges, actually, of applying this kind of technology, is figuring out which genes to mutate and how to mutate them so they actually do something useful. I'm going to switch a little bit and tell you a little bit about Theobroma cacao, also known as the food of the gods or the chocolate tree. Here you see a small tree on the left with a lot of beautiful fruit on it. Cocoa pods inside are the seeds that are fermented and then dried and shipped to the chocolate factories where they're ground up and made into chocolate. And these plants are grown around in the tropical, wet tropics throughout the world, in some of the most poor countries on the planet, but also in some of the most biodiverse countries on the planet. So cacao, has a lot of benefits, not only to the chocolate industry and for consumers, and also some of the farmers, but also benefits to the ecosystem. And you can see here an example of how cacao might be grown underneath some very huge shade trees. This is in Brazil, and if you measure the biodiversity of this cacao farm, it's actually 75% of a natural rainforest. Those big trees, all those big shade trees, they provide habitat, they protect the water, they conserve the soil. So there's a lot of environmental benefits of growing cacao if it's grown properly. And another aspect of this is cacao and coffee, a couple other plants, often are growing very close to some of the most important biodiversity sites. And this is an example here of one, the World Heritage Site that is surrounded by cacao and coffee farms. So we think of the coffee and cacao farms as sort of buffering and protecting this region of high biodiversity. If we didn't have plants like that or farms like that, the next go-to generally would be something like a cattle farm, which has a biodiversity of, just a couple of plants and a couple of animals, and it's not so good as a buffer for rainforests. But even maybe more important is the benefits to society and the farmers. So there are 5 to 6 million cacao farmers in the world that we know of. They generally have very small farms, but it's a very important source of cash. They probably grow other things like their own food. They maybe have jobs in town and they do other things. This is sort of a cash cow that farmers can plant 20 or 100 trees and have a small amount of income for their families. It's also used by development agencies as an alternative to illicit crops, mainly coca in Peru and Colombia. And so people that have been forced to grow illegal crops, generally they would prefer not to do that. And so there's a large number of projects around the world where coca and marijuana and other crops, poppies even, have been replaced by cacao. I already mentioned all the environmental services. So there's a lot of good reasons to grow cacao in addition to supporting a multi-billion-dollar industry. But there's a lot of threats that the farmers face, and it's really difficult for them to be profitable and sustainable. You can see that plant diseases is their major threat, and about 40% of the crop is lost. They also have very low yields, in general 300 to 350 kilograms per hectare per year, where the potential is up to 10 times higher than that. And the main reason that they have such low yields is because they lack really the materials that they would need to control pests and diseases. They don't have access to good fertilizers. Many farmers don't have access to training or knowledge how to take care of their plants. So, this means that a lot of these farmers have very low incomes. In fact, in some areas as low as $350 a year. And that gives a much, it's a bad cycle because then the farmers don't have money to invest, and the younger kids don't want to go into cocoa farming, and the farmers are getting older, and it generates sort of a negative spiral. Just to make matters worse, of course, climate change is already affecting cacao. We see in areas droughts and floods and different changes in temperature. Cacao does pretty good with changes in temperature, but it does not do good with changes in water amounts, especially distribution throughout the year. So it's susceptible to droughts and flood. We're already seeing some problems in certain areas. So you might be wondering why at Penn State, why do we do cocoa research? And so just a little bit of background on that. Way back in 1986, the American Cacao Research Institute started this endowment at Penn State, and it's a permanent endowment that funds the base funding for this program. And as you might guess already, Hershey was one of the major companies that was part of it, but Nestle's and a whole number of other companies were part of this ACRI. And but that's just the base funding for our program. And we get additional funding from various sources, including USDA and the National Science Foundation, as well as private companies. So our general strategy right now is to try to develop the technology and the ability to use these modern methods of crop improvement with cacao. And so in order to do that, we're starting at the basic knowledge. We're studying the genome sequences of these plants. We're trying to discover the important genes and the important traits. And then we're trying to find the versions of those genes or the alleles that confer a beneficial trait. And ultimately, we want to develop markers, DNA markers, that can be used to track those good versions of the genes to accelerate breeding. Breeding a cacao new variety could take 20 or 30 years right now. And we think using modern methods of crop breeding, we could accelerate that to 10 or even less years. That'll generate better varieties, that'll help the farmers with their income, and hopefully lead to sustainable cocoa supply. Or it's one of the things that could contribute to that. There's other aspects of this as well we're not going to talk about, but this is the genetic approach to sustainable cocoa supply. Now, this is pretty complicated. So we're interested in not only yield but quality. Most traits are controlled by many genes, or they're called multigenic traits. These are just some of the interesting traits that can be bred for. For example, can we increase photosynthesis efficiency? There are genes for carbon partitioning. Could we put more of the carbon into the yield part of the plant and less into the non-yield part of the plant? In the case of cacao, that would mean more seeds and less wood, less branches, for example. Genes for fruit development, which is, inside the fruit is the seeds. Drought tolerance, I already mentioned how important that is. Nutrient use efficiency, especially nitrogen and phosphorus, are often rate limiting for production. If you can breed plants that can use limited nutrients better, more efficiently, then you're going to have a better yield. And last but not least, genes for disease resistance, which is what my lab is mainly focused on right now. And so we've all heard about how animals, with our immune system, and we can become resistant to pathogens. Plants have a very similar type of pathways, or very complex immune system. In fact, it shares some relationship and some evolutionary basis with even the human immune system. So parts of this mechanism are conserved through billions of years of evolution. But just a very short, simple version of this shows you that when a pathogen comes to a plant, the plant recognizes the pathogen, and the plant then responds to that pathogen. And it can do so both locally, there's a local response, and there's a distal response. So basically the plant recognizes the presence of a pathogen. It sends signals throughout the local region, throughout the cells that recognize that pathogen, and those cells turn on a lot of genes and a lot of mechanisms, very, very complicated, thousands of genes involved, to try to stop the pathogen. And the plant also can signal, it can put mobile signals that move through the plant. So in distal places where the pathogen hasn't even arrived yet, the plant can start to also respond. We call that the systemic acquired resistance. Very complicated pathways. No time to really go into much detail here, But there's hundreds and hundreds of labs around the world working on discovering and understanding this mechanism, because once we fully understand this mechanism, we're going to be able to breed much better plants for resistance to pathogens. Yeah, the plant responds by doing a lot of things. One thing is the plant can thicken the cell walls. That makes it harder for pathogens to get through. The plant can make a large number of antimicrobial peptides, proteins, metabolites, lots of things that kill pathogens or slow them down. One of those being reactive oxygen species. That's a major, one of the major mechanisms, putting out highly reactive, toxic, reactive oxygen species that can kill cells. Yeah, that's mainly what I wanted to say. So one of the things we're doing in my lab, one of the genes, so basically we're hunting for these important genes for disease resistance. And we found a few of them. I'm just going to talk about two of these, NPR1 and NPR3 that you see here. Basically, when, again, the plant sees the pathogen, there's a signaling cascade. This NPR1 protein moves from the cytoplasm into the nucleus. When it gets into the nucleus, it turns on about 2,000 genes that lead to resistance to the pathogens. Now, the plant uses another protein, NPR3, that basically shuts down this pathway. It's a repressor of the defense response. And the reason is, if you have your defense response constitutively activated, if it's turned on all the time, for example, in a human example of this would be, we get a fever to try to fight certain kinds of infections. You don't want to have a fever all the time, so you have a system that shuts that whole pathway down when it's not needed. So the plant has a similar thing, NPR3. It binds to the NPR1 and basically causes it to be degraded. And when it's degraded, then that shuts off the defense response. So there's a switch here that's an on-off switch for the plant defense response. And we decided that we would study this. We want to understand how these genes, the genetic diversity of these genes, and find versions of these genes that are more effective in agriculture. So one of the things we've been doing is we have sampled cacao plants from around the Amazon basin. This is where cacao evolved, and you can see that's a very large area where there are wild cacao plants. We have sampled those and sequenced their genomes. We have now the sequence of every single gene in a cacao plant from all of these locations. That's about 28,000 genes. And basically, by comparing them to each other, we're starting to find ways to discover the most important candidate genes. And we also look at, of all those samples that we collected around the Amazon, we measured their resistance and susceptibility to Phytophthora palmivora, which is one of the major diseases of cacao. And what you can easily see there is this is basically an index of how susceptible these plants are to this disease. On the left are the worst plants that get infected really badly. On the right are plants that are more resistant. So what we were doing is we took the pink, plants that are in the pink pile and the green pile on the right and the left, and we have sequenced all those genomes. So we have a big contrast between their ability to fight the disease. And that's one of the ways we can get a handle on what are the important genes for disease resistance. So we simply asked the question about this NPR1 and NPR3 gene. If NPR3 shuts off disease resistance, what if we knock out NPR3? Would that allow NPR1 to constitutively activate the defense response? Similar to my analogy, would be giving a human a permanent fever and maybe enhance resistance to pathogens. And I'm not going to go in the details, but you saw how we made the little cuts, the cartoons of the scissors cutting the DNA. Well, here you can actually see a picture of a gene. That's the NPR3 gene. And this is the data that shows that using CRISPR, we are able to cut out this short bit of DNA. It's actually 973 bases of DNA. We cut that out of the genome, and it's repaired back together. It was a perfect. We could find examples of perfect excisions, and we're able to regenerate plants that contain this deletion. So the NPR3 gene was completely destroyed in those plants. And further, what we're able to do in a lab setting is to show on the left, when you put pathogen on to the leaf of a CRISPR edited plant on the very far left, you can just see those little brown dots. You don't have to even look at the data, and compare that to the one on the far right that's highly susceptible to the disease. So in fact, cutting this NPR3 gene did increase the resistance to the pathogen. Now, we have not put these plants into the environment yet, mainly because it's really difficult to do so. I'm sure you're aware there's a lot of resistance to GMO plants, and even though these plants were created using CRISPR, they still contain a transgene. And so they're regulated as transgenic plants. So for now, these exist only in our lab, only for research purposes. But there have been a number of interesting crops that have been already made and either under development, or some of these are actually on the market already. And one of them was created right here at Penn State in Yinong Young's lab. It was actually the anti-browning mushroom. And this was the first CRISPR-edited organism to receive a letter from the USDA that said that this mushroom would not be regulated as a GMO. Well, this is a very big news around the world, actually, and still is. In addition, you can see a high amylopectin corn that was created. That high amylopectin has a low glycemic index. So this is actually a special type of starch that's created in this corn. It has industrial uses, but it can also be used for people that for example, have diabetes. It could be better for them. Another one that was produced is this reduced acrylamide potato. Strangely enough, when potatoes are deep fried, they can, one of the byproducts of the deep fry process is acrylamide. Acrylamide is a neurotoxin, and there are significant amounts of acrylamide can be produced in French fries. And this plant was a simple knockout of a single gene that was part of this pathway, and there's very, very low acrylamide production in those potatoes. But as far as I know right now, this product has not really been a success because of the backlash against GMOs. And the biggest user of these would be McDonald's, and McDonald's has a no GMO policy right now. So this is sort of where science and science disbelievers intersect with policies of companies and can slow down the rollout of a technology. Another beautiful example of this is the reduced trans fat soybean oil, which is basically changing a little bit one of the genes for oil biosynthesis in soybean that led to a type of oil that's healthier for people. And I'm going to leave it at that so we can go on to the questions.
Okay, Mark and Jason, thank you both. We'll go ahead and move on to some of the questions. We've got a few questions that have been submitted in chat and in Q&A, but we'll go to the ones that were originally sent in. A couple of them are pretty broad, but maybe you can give kind of an overview of what you think about them. So one of the questions that was submitted was, how far has this technology been developed? I think you can see from today that it's been developed in quite a lot. Jason, you think there's a lot more room to go?
I do. Again, I tend to be relatively skeptical of a lot of the sort of the gene drive type strategies. I think in terms of the population suppression, I mean, Oxitec has already been doing that all around the world, and it seems to work with minimal sort of issues. In terms of gene drive, particularly for population suppression, but also potentially for population replacement, there's still quite a lot of sort of scientific and more importantly, regulatory work that needs to be done, because these things are built to spread. So if you release them in one country, you can't stop them from going into neighboring areas and things that they may not want that. So there needs to be a regulatory framework in place to deal with that. I also personally don't think a lot of these strategies will work very efficiently. So again, there needs to be a little bit more, I think, study on that, particularly strategies to mitigate the evolution of resistance to these control strategies. And that kind of work is just starting now.
Great. So, Mark, one of the questions was asked in the Q&A was, could CRISPR increase oxygen production in plants? You talked about a few other things like photosynthetic efficiencies and so forth. Could CRISPR do it by itself, or would you have to do something a lot more than CRISPR in order to change photosynthetic or capabilities or oxygen production?
That's a great question. I mean, again, photosynthesis is an extremely multigenic trait, but I think there are possibilities there as we learn more and more about it to change even single amino acids that could help increase the efficiency of photosynthesis. For example, enzymes that might have a higher temperature tolerance to, to climate change. So yeah, I think it does have a potential for that. In addition, I think, now we're seeing advanced methods of CRISPR being developed where multiple genes can be targeted at the same time. And I think really it's a little bit in its infancy. So if you try to imagine what it was like, just 30 years ago in my field, and then try to project that rate of advancement 30 more years out, I think in 30 years we're going to see some pretty amazing abilities to change entire pathways using CRISPR or probably even more precise methods.
That's great. So, Jason, another couple of questions were asked about the mosquitoes. Said, how soon will they be applied outside the laboratory for the massive death rate in Africa? Are there plans to do this in Africa? And I think you've touched on this already, but do you want to be more specific about
the sort of the sex ratio distortion gene-drive mosquitoes? They're trying to get regulatory approval for pilot field releases right now. Again, I don't know this, for certain, because who knows how that's going to turn out. But my guess is we'll probably have at least some sort of a limited type field release or field trial within the next 5 years. I would not be surprised to see that.
That's great. So, Mark, we also have a question from Mark Lewis, who said, where are we in the process the American chestnut trees? Have you followed that one very much?
A little bit, yeah. We have a big project here. It's at Penn State and with some collaborating institutions where they have actually made transgenic chestnuts. And as far as I know, those are in the field trials. The permits for the field trials have been, or in the very final phases of getting approval. So I think the transgenic crops, transgenic chestnuts, which they have shown good resistance in greenhouse and lab tests, soon will be in field trials. But, it seems like a very long drawn out process because every time they make one step forward, then somebody will file a complaint for another environmental review, and it's been vetted very, very heavily. So, I think those plants seem to work in the lab, but haven't, again, like our cacao trees, haven't really been tested in the field yet.
I think we have some that have been planted outdoors at our extension center at Northeast up in Erie County. But I think
right, they're in those controlled field.
Yeah.
Some of those are outside, that's true, but they're not, what they're planning to do, and they have asked for permission to do, is like distributing them wild in the forest.
Yeah. So there's current proposal to deregulate them, but they look very impressive. And we have a classical breeding program on campus as well. It relied on backcross and Chinese chestnut generations ago and have been backcrossed and so forth. And those are growing out behind the arboretum, and they're not GM, but there's a couple of going.
I think, just jump in for one more second. I think it's important to say, and everybody understand that, there's a lot of classical breeding still going on, and a lot of what we're doing is to support classical breeding, including we use transgenic plants or CRISPR to study the function of genes but that may support in the future just classical breeding.
Yeah. Um, so how do you manage unintended consequences of gene manipulated research outcomes? What do you think is the best pathway forward for additional gene manipulated foods? Mark, you want to try that, take a crack?
Whoa, that could be a whole course really to talk about that. Yeah, there's been a couple of examples of unintended consequences, negative consequences of both conventional plant breeding and GMOs. One of the famous ones was right here at Penn State, the Lenape potato that had high amounts of toxin in it, and it actually got launched as a product until it got to enough people and they realized it was making people sick. But that was just conventional breeding. There is an example of this where a allergen was put into a crop that was meant to be released. It went through the regulatory approval process and was not released. So, so far, there's not one documented example of any human being hurt by any transgenic crop. And that's trillions of meals, both to humans and animals. However, there is a always a potential for some problems. So mostly it's regulated right now by USDA, FDA, and they have a whole process of approvals to release transgenic crops. It's a pretty transparent, like you can go to the website and learn about how exactly what you have to do. Lots and lots of tests need to be done. But really the onus is on those companies. So I think the companies are the ones that have to do all the safety testing and be sure their products are safe. No company wants to release a product that's going to hurt people because that will be the end of their company. So, there's a lot of feedback loops for people to be very careful. The way this is done, the concept is substantial equivalency. Basically what we do is to study a new transgenic crop and compare it to the predecessor, the non-transgenic crop. And they tear it apart to every, degree that you could. What are all the molecules in there? What are the concentrations? Did the transgene create any big changes that we didn't anticipate or anything that could be a problem? So it's a lot of science that goes in and a lot of very powerful approaches are used to study those plants before they would be released.
So, Jason, Nancy Kraft has asked questions. She's asked, is CRISPR only used in plants or also in animals? And you show this being used in insects and other arthropods. Is CRISPR used in aquaculture? Has anybody done this for salmon or something like that?
Definitely for salmon. And then there's a company called Aqua Bounty. That's sort of their whole deal is genetically modified salmon. There's also been a number of people that are trying to get CRISPR to work in other sort of aquatic organisms. Bobtail squid was just done last year, which is sort of a model system for things. It was one of the first cephalopods to be genetically engineered. And again, the major issue with applying it to a lot of these species is not that the CRISPR doesn't work, because it does. It's basically introducing it into the eggs and getting it to where it needs to be to result in heritable germline editing. And that's actually why we developed some of the technology that we've developed in my lab, is to make those kind of things easier. And so we've actually been working with people working in fish and squids and coral and other things, and trying to adapt some of our processes into those novel systems. Definitely, in terms of things that might enter the food chain, salmon is probably the biggest one right now.
Right.
Also trout.
Yep. Mark, Daryl Farber asked about acquired resistance similar to induced resistance.
Yeah, those are two sort of overlapping complex mechanisms that intersect with each other. But induced resistance is when the pathogen basically triggers a pathway inside of a cell, and then systemic acquired resistance is generally thought of as that signal then moves through the plant and the rest of the plant becomes resistant. And again, the pathways to that resistance often are overlapping sets.
let's see. Lorraine Minio asked about is CRISPR being used to increase the CO2 uptake in plants?
Again, yeah, I think that is a possibility. I know there's a lot of labs, I don't work in that field, but trying to increase photosynthesis efficiency will just by nature increase the amount of CO2 uptake of a plant. And CO2 uptake underlies a lot of the physiology, obviously, of the plant. So anything that would increase the yield or the growth rate of the plant's going to increase CO2 uptake. But specifically looking at CO2, there are projects underway to look at that reaction mechanism where CO2 is taken up and trying to improve, say, the efficiency of those enzymes. And I think we actually have a person in the biochemistry BMB department here at Penn State who works exactly on that.
Sarah's asked a question, what mechanism does Penn State have in place to monitor or regulate the research? I'm sure that both Jason and Mark Ved go through a lot of this. You want to give a bit of a rundown?
Sure, Jason.
Yeah. So we run into this all the time. Any kind of research, if it's involves recombinant DNA technology or pathogen work or animals, is all covered by various regulatory committees and approvals before you can start the work. And so you have to write a protocol, and it goes to the committee, which is made up of experts on campus and also members of the public for approval, and it can go back and forth. I've had protocols that have taken many, many months to get approved because the committee wanted to make sure everything was just right. So it's not a trivial thing. You can't just sort of go in and just do experiments. You have to actually get regulatory approval for all of it, which is pretty rigorous in terms of the things that you need to do in order to get approval. Without going into specific details, but it's quite a lot of work actually to get it done.
Yeah, it's the Office of Environmental Health and Safety. In addition to that, we also work with the USDA APHIS, the Animal Plant Health Inspection Service, I think that stands for, and they regulate how we import plant material or export it for that matter, and some of those go into quarantine. We have a quarantine greenhouse section now that can control and try to make sure that no pathogens or insects and things could get released from the greenhouse. All these are under the National Institute of Health has a huge guidelines for recombinant DNA, and that covers plants as well as cloning of DNA and other kinds of this technology. And we have to follow all those guidelines. Those guidelines also gradate. There's biological safety levels 1 through 4, I think is the top one, whereas 1 is almost nothing but basic lab safety protocol.
Yeah. So we're running close to end of time. There's another couple of questions that come in beforehand about risk of GM crops and so forth. And I just want to draw your attention to, there was a issue of, let's see, just a second here. There was I hope everybody can see the slide of the purple tomato, the indigo tomato. This is in the New York Times, just in July 20th. They had a whole insert in the Sunday Times about learning to love GMOs, where they argue that the fears have turned the public against genetically modified food, but potential benefits have never been greater. And it just shows this example of how you could take a tomato and add even more antioxidants to it than tomatoes can be done normally. So there's a lot out there in the popular press that you can follow up on this if you like. In any case, we've just about run out of time. I want to extend a special thank you to Professors Jason Raskin and Mark Guiltinan for taking the time today to share with us and give us insight into what they're doing. Please note that the next session will be listed on your screen shortly. I'm going to stop the share and give Alan a chance to put up the next session. It's set for October 20th, where I'll be joined by Rob Bertram, who's the chief scientist with the USA Agency for International Development's Bureau for Resilience and Security. as well as Dr. Deanna Beering, who's with us at the College of Agricultural Sciences, Assistant Dean and Director of Ag Sciences Global. So thank you very much for joining us today, and we hope you've had a great time with us. I've definitely enjoyed the conversation myself. Thank you all, and until next time, best wishes from College Connections.